Jumping technique and climbing behavior of the columbolan Ceratophyzella cingulata, columbola, hypogastruridae. Zool. 3. CH3012 Bern, Switzerland, email, Jurg.zettel. Keywords. Columbola, hypogastruridae, Ceratophyzella cingulata, jumping, orientation, climbing behavior, migration, eversible vesicles, polymorphism. Abstract. Ceratophyzella cingulata uses protrusible vesicles on its antennae and posterior end as sticky landing devices to avoid tumbling upon landing. This technique facilitates its orientated movement during winter migration. It allows the animal to use directional jumps without rebounding even when climbing tree trunks. The antennal vesicles are present only in the surface active morphs of C. cingulata, and only these individuals jump readily. The vesicles are everted only when the animal has time to prepare the jump. In an escape leap, they are not protruded and therefore the animal tumbles upon landing. Introduction. In columbola, the usual way to move is by crawling, but jumping is used as a very efficient mechanism to escape from attack or disturbance. Details on this extremely rapid movement have been given by Bauer and Wallenkel, 1976, and Christian, 1978, 1979. Jumps are energy-consuming, and after a few repeated leaps the individual springtail has used its available ATP and remains immobilized until the energy reserves have been regenerated. Rufus and Zinkeler, 1995, personal observations. Winter active columbola jump regularly when migrating. This behavior has been described for Hypogastrura socialis Lifford, 1975, Linaz, 1981a, Hagver, 1995, for H. laponica, Linaz, 1981b, and we have found it in all the species we observed during winter activity so far. Hypogastrura socialis, Ceratophyzella cingulata, Vertagopus westerlandi, Isotoma himalis, Elsotomerus schaeferi, Agrenict bidenticulata. By strictly orientated jumping, columbola are able to cover remarkable distances within a short time. In C. cingulata we observed up to 3 meters per hour, and Hagver 1995 calculated up to 200 to 300 m per day for H. socialis. Several authors report on the ability of columbola for visual orientation. Muller-Krainer, 1990, Hagver, 1995, own observations. As springtails rotate during their leaps and land at high speed, they tumble upon landing and it takes them some time before being able to move on in a specific direction. The present paper describes the jumping behavior in Ceratophyzella cingulata, which is able to avoid tumbling by a special technique, being not only of significance during migration, but also for feeding behavior. Material and methods. Ceratophyzella cingulata, Uzel, is a winter-active hypogastrurid known from the northern forelands of the Alps Palisa, 1994, but also from North America. Babinko et al. 1964 Establishedsi pseudoarmata Folsom, 1916, to be a junior synonym of C. cingulata. First observations on its biology were made by Zettel and Zettel, 1994a, b. The columbolan shows an unusual migration behavior during winter, with dense colonies comprising up to hundreds of millions of individuals moving over the surface in the same direction. Field observations. Since 1990 we have investigated this species in a mixed forest 10 kilometers north of Bern, at an altitude of 640 meters A.S.L. The present data are part of a program covering the migration patterns of this species. Individual jumping behavior was observed either with a short focus telescope, magnification x8, on the soil surface, at a distance of 1.5 to 2 meters, or with a dissecting microscope, magnification XL4, climbing on trees, at a distance of ca. 20 centimeters, both instruments being mounted on a tripod. No artificial illumination was necessary for both types of observation, and the animals did not seem to be influenced by the presence of the observers. Observations in the laboratory. Some aspects of the behavior had to be investigated in the laboratory, using a dissecting microscope with a magnification up to x40. The animals were kept in climatic chambers and observed directly in the culture vessels, glass vials of a diameter of 8 centimeters with a moist base layer of plaster of Paris. Observations concerning orientation and jumping distance were carried out in a climatic chamber at field temperature, 0-5 degrees Celsius. Individuals were released from a culture vessel onto a big sheet of rough paper and their positions were recorded directly by tracing their displacements with a pencil. The marking of starting and landing points as well as positions at intervals of 10 seconds during crawling could be done at a distance of 1 centimeter behind the animals without disturbing them. To exclude an orientation on landmarks, the animals had to move in an arena surrounded by black paper. The light source was positioned either on one side of the arena or directly above it, producing a diffuse illumination against the ceiling. Photography in the laboratory. To analyze the sequences of takeoff and landing, we took photographs with a Leitz photomicroscope, magnification up to *32, and a high-speed flash 1/8,000th of a second. All pictures were taken in a climatic chamber at the actual culture temperature of 5-10 degrees Celsius. It proved much easier to take several single shots to obtain pictures of all positions in the sequence than to use a high-resolution stroboscopic flash or an ultraspeed film camera, because we needed pictures of undisturbed and spontaneously jumping animals. The takeoff was recorded from the tip of a short vertical glass capillary into which some animals had been dropped. They climbed up, and when emerging at the tip of the capillary, many of them prepared to jump. By illuminating from one side, they could be guided in the desired direction. Landing was either recorded frontally through a vertically mounted microscope cover slide, against which the jumps were directed by the lighting, or from an angle of ca. 45 degrees from above in the culture vessels. Shutter and flash were triggered manually when an animal appeared within a certain frame. Among ca. 200 pictures taken, 1/3 was successful, i.e. provided photographs of all positions needed for the interpretation of the extremely rapid movements during takeoff and landing. During the preparation for the jump, it was easy to trigger the shutter at the appropriate moment, because this sequence lasted at least 0.5 s. To record the landing, several hundred individuals could be made to simultaneously start to climb the glass wall of the culture vessel, orientating towards the light source outside the jar. By this method, we often photographed several different positions among 30 to 50 animals in the focus plane. Results. Jumping morphs. C. Sigillata passes through 4 morphs within 1 year of its life. For details see Zettel and Zettel, 1994b, but only 2 of them are surface active. One of these can be observed in December, January and February, March, the other in April, May. During favorable humidity conditions, the winter active morph climbs onto trees to feed on coccolalgae growing on the trunks. Only the 2 surface active morphs jump readily, the other 2 which remain in the litter or soil show jumping only as an escape behavior. As a typical hypogasterid, C. sigillata has eversible anal vesicles. An additional and important feature of the surface active morphs is an eversible vesicle between the antennal segments 3 and 4, being a characteristic of the genus Ceratophysella, which is lacking in the soil-dwelling morphs. These eversible parts prove to be of central significance in the jumping sequence. Orientation and jumping frequency. Figs 1 and 2 show that a light source can be used for orientation. With a distinct light source, most animals moved in a strongly directional way which was not necessarily orientated towards the light source as it was the case in Figure 1. The majority of the animals used in an experiment moved in the same direction. Under a diffuse illumination, no linear trajectories could be observed. With a visible light source, jumping was significantly more frequent than under diffuse lighting when C. sigillata showed no orientation, p less than 0.001% 2 test. A mean jump measured 54 plus or minus 0.5 mm, mean plus or minus 95% CI. And the leaps tended to be shorter without an orientation possibility. As the collembola do not land on their feet, they had to grip the substratum and reorientate after landing. They orientated by turning on the spot until their body axis was in the same direction as the preceding jumps, sometimes combined with some short crawling. In the field, a significant difference between the reorientation times in colonies being mainly resident or during migrating was observed. In moving colonies, the mean time between 2 jumps varied between 10.1 plus or minus 1.06 and 11.1 plus or minus 2.1 s, n equals 50, mean plus or minus 95% CI. In resident aggregations, the time was between 16.9 plus or minus 1.80 and 18.9 plus or minus 2.37 s. Animals not aggregated in dense colonies spent more time crawling, 2 leaps being separated by 91.5 plus or minus 71.95 s, n equals 8, Egger, 1998. Preparation for jumping. There is a clear difference between a spontaneous takeoff, when migrating, or when disturbed. Whenever possible, the animals prepare for a jump. This lasts at least 0.5 seconds. 3 steps can be distinguished during this phase, Figs 3, 4. As soon as an animal has decided in which direction to jump, it takes a firm grip with its feet on the substratum and contracts its body muscles, resulting in a visible shortening of the body. By increased her. Figs 1, 2. Trajectories of migrating C. sigillata in a climatic chamber at 5 degrees Celsius. Positions are marked at intervals of 10 s and jumps displayed with bold lines. 1. Strictly directional movements with frequent jumping under direct illumination from one side, arrow. 2. Disorientated movements with reduced jumping under diffuse light conditions. Figs 3 to 7. All figures are drawn from microphotographs. Everted parts, vesicles, ventral tube, are stippled. 3. Preparation for takeoff in C. sigillata. The antennal vesicles are fully protruded, the abdomen is already contracted, but the anal vesicles are not visible. 4. Comparison of the normal attitude of C. sigillata with an individual ready to jump. All vesicles protruded. 5. An individual of C. sigillata in full flight, immediately before hitting a vertical surface. Due to the high internal body pressure, all eversible vesicles in the ventral tube are protruded, legs and furca fully spread. 6. C. sigillata after landing head first. The animal sticks to the glass wall with its right antennal vesicle, moving with its legs. Internal body pressure is already normal, anal vesicles withdrawn and furca flexed. 7. C. sigillata after hitting the glass wall with its posterior end first. It hangs on the anal vesicles, moving with its legs, antennal vesicles already withdrawn and furca flexed. Gore, the eversible antennal vesicles are protruded, and the tips of the antennae bent upwards and inwards. In the final step, the internal pressure is further increased and the anal vesicles as well as the ventral tube are everted. These parts remain protruded during the entire leap, Figure 5. When disturbed, the animals are able to take off without preparation within milliseconds, but in this case neither antennal nor anal vesicles are protruded. Landing. The difference between these 2 jumping behaviors becomes obvious upon landing. After an escape leap, the landing is the same as after any collembolan jump. Due to the enormously high speed, the animal is tumbling and has to struggle to get onto its feet again. After a prepared leap, the animal sticks to the substratum and does not tumble. Due to their wet surface, the protruded vesicles act as sticking devices. A landing collembolan can hit the ground directly either with its antennae or its posterior end, Fig 6, 7. In such cases it sticks to the surface and by bending its body and moving its legs it regains its feet and continues directional movement. When landing on its ventral or dorsal side, the body continues to rotate in the direction of the leap and there is immediately contact between the sticky vesicles and the surface. On a horizontal surface cardboard exposed in the field, 30% of the landing animals became stuck with the antennae, 20% with the anal vesicles, 50% landed with another part of the body first. On a vertical obstacle placed in the way of a Figure 8. C. Cigillella crowding on the tip of a bark flake when climbing a spruce trunk. Migrating colony, 60% landed head first and stuck with their antennal vesicles, 40% stuck with their anal vesicles. Egger, 1998. Jumping during climbing. During surface activity and when the tree trunks are moist, C. cigillata often climbs to several meters in height, to feed on cockle algae growing on the trunks. Suter, 1993. There is a difference when climbing a beech or a spruce trunk. The bark of beech, Fagus sylvatica, is smooth and the climbing springtails move continuously upwards without being hindered by obstacles. The trunk of spruce, Picea abies, is covered by projecting bark flakes. Each flake is a dead end for climbing collembola, which leads to crowding at the edge of each flake as the animals attempt to escape from it. Figure 8. When the crevice behind the flake is deeper than a few millimeters, a few individuals climb down on the rear side of the flake, but most jump towards the bark behind the flake, where they stick with their antennal vesicles without rebound and continue to climb upwards until the next flake is reached. Egger, 1998. Discussion. Jumping in collembola. Up to now, investigations of springtail jumping have been carried out by individuals being forced to jump by a mechanical or chemical stimulus. In such cases, the jump can be predicted precisely, allowing the use of high-speed cinematography. In undisturbed individuals, takeoff may occur anytime within several seconds after the first signs of preparation. Therefore, we chose an alternative recording technique to analyze the preparation for a jump as well as the takeoff and landing. From High-Speed Cinematographic Pictures, Christian, 1979, Brackenberry and Hunt, 1993, and the models by Brackenberry and Hunt, 1993, it can be seen that animals with a long furca perform steep and often backward-orientated somersaults, but species with a short furca like the hypogastrurids show relatively low trajectories with a much slower rotation. It is interesting to realize that only the surface active morphs of C. cigillata jump spontaneously and only these individuals have antennal vesicles. The morphs remaining in the soil lack antennal vesicles and do not jump spontaneously. Orientation. The laboratory experiments showed that C. cigillata is able to orientate with the aid of a light source. The chosen direction was not obligatorily towards the light and could chance with time. However, orientation during migration cannot be based only on such a mechanism, otherwise the animals would change their direction continuously during the day according to the sun. This was not the case. Migrating colonies often kept their direction for several days. Nothing is known about how springtails are able to compensate for the movement of the sun, as is known for some insects. Observations by Hagver, 1995, suggest that course patterns in the environment can be used for orientation, and that a directional movement is also possible without sun. We never observed a deorientation of C. cigillata under an overcast sky. It is possible that C. cigillata is able to use other cues in addition to visible light. Muller-Krainer, 1990, showed that light-dark patterns were used for visual orientation in Entomobria corticalis to enable movement towards tree trunks. Our observations in the forest indicated that C. cigillata was able to perceive a tree trunk visually at a distance sufficient to avoid it during migration. However, as also Muller-Krainer, 1990, demonstrated in his species, this visual cue had to be greater than a certain minimum height. Migrating colonies did not jam in front of a tree, but regularly against low obstacles like roots or branches lying on the ground. These could not be discerned at distance and were too high to overcome with a single leap. Such an orientation facility does not indicate anything about visual orientation during climbing, when the animals head towards the next bark flake. There is no precise orientation needed, because it is enough when they head towards the dark bark in front of them. They need not to estimate any distance for a safe landing, because their body rolls on landing and this guarantees contact of their sticky vesicles with the substrate before they rebound or fall. Climbing onto trees. On favorable days, almost the entire population can be found on the trees. Egley and Carnke, 1996. The trunks appear to be covered with purple-gray paint, due to the springtails colonizing the bark in densities up to 200 Ind, CRN2 and being in body contact with each other. When other springtail species jump during climbing, they shoot themselves of the surface and fall down to the ground. Even when coming into contact with the bark again, they will rebound. The situation is completely different in C. cigillata, which is able to jump successfully during climbing. The sticky vesicles avoid a fall, either by immediate contact with the bark or after a short roll. During all the experiments and observations in the climatic chamber as well as in the field, none of the springtails fell or rebounded, even when hitting a slippery vertical surface, glass or plastic, wet bark. Christian, 1979, demonstrated that the starting angle for Hypogastrura socialis was 77 degrees. When jumping normally, C. cigillata performs leaps of similar height. However, from the tip of a bark flake or from a capillary in the laboratory, the animals always used a very low trajectory, i.e. for the shortest way out of the dead end. The starting angle in such cases was much lower than when taking off from a horizontal surface, this being attained by a stooped attitude of the body. For distances of 10 to 20 millimeters, as they mostly occur in such situations, a maximum of 1 to 1.5 rotations can be calculated from the data given by Christian, 1979, for H. socialis. There is no choice of distance possible, but as mentioned above, the animals only need to head for the nearest vertical structure. In most cases they will hit the surface with their antennae first, but sometimes we also saw animals sticking with their anal vesicles. With this special jumping technique, C. sigillata is able to exploit the algal growth more easily on spruce trunks, which is of importance in increasing their cold hardiness, because they sequestrate antifreezes from the algae J. zettel, C. suitor and S. strike, in prep. Significance of vesicles. Linnaeus 1988 described anal vesicles from a number of peduromorph collembola. In H. socialis, they have the form of finger-like tubes. In other species, e.g. in C. sigillata, they are much shorter. Linnaeus thought they were of significance for jumping, as he saw them being everted before takeoff. As we now know, they are everted just by the increased internal pressure when preparing a jump. He also suggested an adaptive value of the anal vesicles, connected with pheromone release in readily jumping springtails. In such species the vesicles are much longer than in those without a distinct jumping behavior. Linnaeus, 1983. As can be seen in the photographs published by Christian, 1979, disturbed H. socialis do not show protruded anal vesicles. Eversible antennal vesicles are not present in Hypogasnera species. His animals were stimulated to jump and therefore did not prepare to jump as seen in migrating animals. In fact, the animals rebounded when hitting an obstacle in his experiments. Christian also mentioned that spontaneously jumping H. socialis often stuck to moist surfaces when jumping, but he offered no explanation for this phenomenon. The ventral tube, which is also protruded during a leap, does not play a role as a sticking device in hypogastrurids, because it is too short to make contact with the surface. However, the ventral tube can be used as a climbing aid on slippery surfaces, a behavior which is easily observed in culture vials. The situation during jumping can be quite different in smintherids. Brackenberry, 1990, described Smintherus voridis using its long ventral tube to ride in the body after a jump. Christian, 1979, mentioned that in S. voridis the ventral tube is everted during a jump and with its moist surface might play an important role as a sticky landing aid within vegetation. An adaptive value of protrusible vesicles can be postulated in migrating collembola from the point of view of the more sophisticated landing technique. Reducing in tumbling in regularly jumping species may be a mechanism to reduce time and energy spent during migration, especially when suitable environmental conditions prevail. Gate current research on the impacts of urbanization. Gate current research on the impacts of urbanization using odonats as a model group. First, we will present a brief overview on the impacts of urbanization on terrestrial and freshwater ecosystems. Then we conduct a systematic literature review regarding odonats in urban environments and outline the plausible effects on odonats, followed by the use of odonats as bioindicators, the conservation value of urban wetlands for odonats, a summary of evolutionary strategies for species to cope with urban stressors, and finally we conclude by identifying research needs. The effects of urban stressors on terrestrial and freshwater ecosystems. The wide range of impacts associated with urbanization indicates that it exerts a considerable effect on terrestrial biodiversity. Most studies show that species richness and evenness is reduced in highly urbanized regions, depending on the taxonomic group observed, degree of urbanization, and spatial scale of analysis. McKinney, 2002, 2008. For example, butterflies Rushchick, 1987, Rushchick and D'Arajo, 1992, Blair and Lawner, 1997, and ground arthropods McIntyre et al., 2001 show this tendency. The general pattern of biodiversity decrease in cities is probably due to the fact that over 80% of land in city cores is covered by buildings and pavements, with the remain. Dragonflies and damselflies Odonata in urban ecosystems. A review. G. Iavana. Odonata, urban ecology, city, anthropogenic stressors. Abstract. The expansion of urban areas is one of the most significant anthropogenic impacts on the natural landscape. Due to their sensitivity to stressors in both aquatic and terrestrial habitats, dragonflies and damselflies, the Odonata may provide insights into the effects of urbanization on biodiversity. However, while knowledge about the impacts of urbanization on odonats is growing, there has not been a comprehensive review of this body of literature until now. This is the first systematic literature review conducted to evaluate both the quantity and topics of research conducted on odonats in urban ecosystems. From this research, 79 peer-reviewed papers were identified, the vast majority, 89.87%, of which related to studies of changing patterns of biodiversity in urban odonat communities. From the papers regarding biodiversity changes, 31 were performed in an urban-rural gradient and 21 of these reported lower diversity towards built-up city cores. 12 of the cases of biodiversity loss were directly related to the concentrations of pollutants in the water. Other studies found higher concentrations of pollutants in odonats from built-up catchments and suggested that odonats such as Eschina juncea and Platycnemis pennips may be candidate indicators for particular contaminants. We conclude by identifying current research needs, which include the need for more studies regarding behavioral ecology and life history traits in response to urbanization, and a need to investigate the mechanisms behind diversity trends beyond pollution. Introduction. Urbanization is one of the main drivers of ecosystem change. The expansion of urban areas is a main cause of many environmental stressors. I. Removal of native vegetation leading to degradation and fragmentation of the natural landscape. E. Modification of hydrological systems through water extraction for human use. E. Increase of local minimum temperature creating a heat island effect. And IV. Accumulation of pollutants to a point at which the environment cannot fully process and degrade them, causing eutrophication and affecting biogeochemical cycles. Grimm et al. 2008. McDonald, 2008. Moreover, the estimated percentage of the world population living in urban areas now exceeds 50%. Grimm et al. 2008, and almost 60% will be living in urban areas by the year 2030, Seto et al. 2012, Gunaralp and Seto, 2013. Given the range of impacts and rapid growth of urban development, it is essential that we understand in depth the implications for natural systems both in and around cities. Dragonflies and damselflies, Theodonata, have been suggested as barometers for environmental change due to their sensitivity to other anthropogenic stressors such as climate change, Hassel. 2015, and variation in habitat quality, Clark and Samways. 1996. However, the impacts of urbanization on odonats remain poorly understood. In this review, we will investigate. J. Entomology. Sensitivity to a range of urban stressors means that odonate communities have the potential to act as barometers of environmental change in urban areas, as has been demonstrated successfully in South Africa for habitat quality, Clark and Samways, 1996, and as has been suggested for climate change, Hassel, 2015. In this review we outline the stressors associated with urban environments that have the potential to affect odonats considerably across their life cycle. Dragonflies and damselflies is a model system. Dragonflies and damselflies, the Odonata, are highly suitable model organisms for the study of urban ecosystems because they are sensitive to different stressors, such as pollutants. Ferreras-Romero et al. 2009, and temperature changes, Hassel and Thompson, 2008, therefore can be a powerful tool to assess the general conditions of the city environment. B. They are aquatic as larvae and terrestrial as adults, hence can be used as bioindicators in both aquatic and terrestrial habitats. Ortley. 2008, C. They have an important role as predators, hence have a wide range of interactions with different organisms in both aquatic and terrestrial ecosystems. Knight et al. 2005, D. They are ideal for studying movement through the landscape, as their adult stage exhibits a high dispersal ability and are very conspicuous. Conrad et al. 1999, and E. Their biodiversity, ecology and evolutionary biology is well documented, providing a robust foundation for drawing general conclusions. Córdoba Aguilar. 2008. Literature review. We conducted a literature review of the prior work regarding urbanization and odonats, in order to evaluate the potential of odonats as a model system for studying urbanization. During January-April 2014, we used standardized search terms with 3 online databases, Web of Science, Scopus and Science Direct. The words, urban, and, Odonata or dragonfly or damselfly were searched in title, keywords and abstract, except in Web of Science, where the search was in topic. Only peer-reviewed papers including odonats in an urban environment were selected, reviews were excluded. According to the nature of the study, each paper was classified into 1, 2 or 3 of the following categories. Diversity, variables considered here were taxa richness, abundance, probability of occurrence for creating habitat suitability models, diversity indices such as Shannon-Weiner, etc. Toxicology, such as physiochemical features of the water bodies and heavy metal concentration, behavioral ecology, dispersal, trophic dynamics, and life history traits, reproduction, life growth. We went on to examine the references from the papers found in the standardized search and also added those to the analysis where appropriate. The search returned a total of 79 papers, most of which, 65.82%, were published after 2004, see Figure 1, and Table S1 for a complete list of papers with categories. We were also interested in analyzing geographical patterns in Ray. In area used for lawns, trees, and shrubs. Blair and Lawner, 1997, thus fragmenting the landscape and homogenizing vegetation composition. Blair and Lawner, 1997, Feith et al. 2011, Feith et al. 2012, McKinney, 2002. However, plant biodiversity peaks at intermediate levels of urbanization, perhaps due to the high numbers of species associated with suburban gardens. McKinney, 2008. Urban freshwaters have not been explored as much as terrestrial urban ecosystems, despite the high degree of vulnerability that freshwaters exhibit to the pressures of urbanization. Paul and Meyer, 2001, Dudgeon et al., 2006. Urban streams, for example, are usually modified to carry out stormwater into natural streams, washing away industrial and human wastes, as well as road runoff. For man, 2014. Streams are subjected to toxins, temperature change, siltation, organic pollutants, and the replacement of riparian vegetation and substrate for rocks or concrete and, consequently, insects, mollusks, crustaceans, and annelid tend to have decreased species richness and abundance. Paul and Meyer, 2001, Foreman, 2008, 2014, Vaughn and Ormerod, 2012. The presence of organic pollution has been associated with increased abundance of chironomids and oligochaetes, making them the dominant members of urbanstream communities. Campbell, 1978, Seeger and Abrahams, 1990, Paul and Meyer, 2001. Ponds are very frequent in parks and gardens for aesthetic purposes and to improve human well-being. Foreman, 2014, Hassel, 2014, and are greatly influenced by a variety of factors such as the land use of the surroundings, the runoff to the pond, subsurface groundwater flow, and shoreline conditions. Foreman, 2014. Parks and gardens are heavily managed and fertilizers are commonly used, and due to their limited capacity for the processing of pollutants and nutrients, ponds often suffer from eutrophication with an accompanying decline in biodiversity. Foreman, 2014. However, the relative isolation of urban ponds generates a substantial degree of habitat heterogeneity. Each pond possesses its own physical, chemical, and hydrological features, facilitating the presence of a range of species of specialized habitat requirements, hence increasing the differentiation among these ecosystems and the landscape diversity, beta and gamma diversity, respectively. De Meester et al., 2005. They also work as stepping stones, thus facilitating landscape connectivity and potentially acting as metapopulations. De Meester et al., 2005, Hassel, 2014. The biodiversity of odonats in urban areas is expected to be different from their rural counterparts, as is the case for other taxa. However, as already discussed, urbanization is a complex process that encompasses a wide range of selective pressures, and each of them is expected to have a different impact on odonats according to the species and the life stages. Sensitive odonate species may be severely affected by stressors such as contaminants from sewage input, but generalist species are expected to tolerate these conditions, leading to decreased species richness. Moreover, generalist species may exploit the resources available due to decreased interspecific competition, resulting in high abundance of tolerant species. This variation in search regarding odonats in urban ecosystems and found most studies were conducted in United States and Brazil, though plenty were from Austria, South Africa, Germany, Japan, and others. See Figure 2. Of the 79 papers found, 71, 89.87%, investigated patterns in biodiversity, of which 31 were executed in an urban-rural gradient or compared diversity in streams before and after the water flow reached an urbanized area. Of the studies regarding biodiversity in an urban-rural gradient, 21 reported less diversity in cities, and 12 cases of diversity loss were related to pollutant concentration in the water. See Table 1. Below, we discuss the general trend within these papers and give example studies to illustrate those trends. A comprehensive list of studies and summaries of findings can be found in the supplementary information, Table S1. The effects of urban stressors on odonats. Our results suggest that increasing urbanization seems to affect odonate diversity negatively, on the whole, as has been shown in other groups. McKinney, 2008. However, urbanization is a multifaceted problem and the studies reviewed herein offer some perspectives on which aspects of urbanization are having the greatest impacts on odonata across their life cycle and how this may reflect the biodiversity loss in cities. See Figure 3. It is worth mentioning that the stressors listed may not only have a considerable impact in a given stage of their life cycle, but the effects can also be transferred to other stages via carryover effects, e.g. maternal effects from adult to larva, or persistence of impacts between life history stages from larva to adult, except when a decoupling mechanism interferes. For a detailed review, see Stocks and Cordova Aguilar, 2012. Fragmentation. Fragmented landscapes with little vegetation cover and few water bodies may not provide sufficient corridors to facilitate dispersal, thus limiting odonate connectivity within cities. Shavanik et al., 2000, Watts et al., 2004, Sato et al., 2008. Urban landscapes were found to function as barriers for Paracertian calamorum, Ischnera senegalensis, Andi, Asiatica in Japan when the genetic differentiation among populations was analyzed. Sato et al., 2008, showing that the effective fragmentation in urban areas is consistent within at least 3 zygopteran species. These results are also consistent between methods of dispersal analysis. Watts et al., 2004 provide evidence from genetic techniques and a mark release recapture study that urban areas cause a strong negative effect in the dispersal of co-Nagrian mercurial in UK. Although did not find any studies using odonates linking habitat selection and urban landscapes. Fragmentation may also affect habitat selection negatively by constraining access to optimal sites, as has been shown in a study using 2 model species representing territorial animals. Van Langeveld, 2015. Vegetation removal and or modification. Increased vegetation cover and biodiversity of plants was associated with increased odonate richness and overall evenness in Austria. Shavanik et al., 2002, Germany. Figure 1. Gortson and Sulling, 2013, France. Jean-Mugan et al., 2014, and South Africa. Samways and Stadler, 1996, Pryke and Samways, 2009, among others. For example, Gortson and Sulling, 2013, studied odonate diversity patterns in ponds across an urban-rural gradient using a multivariate approach and found that vegetation was not only one of the major drivers of alpha diversity, but also trampling vegetation had a significant negative effect. Other studies have reported similar results, but suggested percentage cover off submerged macrophytes was the main component of vegetation affecting odonate diversity. Jean-Mugan et al., 2014. Additionally, indigenous plant species have also been highlighted as a key factor shaping odonate communicius in urban wetlands, and leaving a strip of indigenous riparian vegetation of at least 20 meters between the stream edge and commercial plantations seems to facilitate the presence of sensitive odonate species such as Chlorolestes tessellatus in South Africa. Samways and Stadler, 1996. The strong management of vegetation in terrestrial ecosystems in cities has a considerable impact on the adult phase of odonates. Silva et al., 2010 because vegetation is one of the main cues used for habitat selection and it influences a vast range of behaviors such as basking, foraging, roosting, sheltering, among others. Buchwald, 1992. Removal or modification of aquatic vegetation from urban freshwater habitats also might have a considerable effect once behavior of odonats not only on the adult phase, but throughout their life cycle. Adults use aquatic vegetation as an oviposition substrate. Buchwald, 1992, often by inserting eggs into submerged plant stems. Corbett, 1999. Once the eggs hatch, the larvae use the submerged plant stems for perching, hiding from predators, and for sit and white ambush hunting. Buchwald, 1992. During emergence, the larva climbs upwards out of the water along vertical plant stems and proceeds to full metamorphosis once it leaves the water. The composition of plant communities may also exclude some specialist species that rely on particular plants, such as Echinophoridis that only oviposits on Strachiotsaloids. Dijkstra, 2006. However, it is worth mentioning that sensitivity to aquatic vegetation loss may vary across odonats, since not all species require aquatic vegetation for oviposition. Sewage discharge and stormwater runoff. Sewage is formed primarily of domestic, commercial, and industrial waste. Wastewater is collected through the sewage system and treated before releasing into freshwater. However, sewer overflows are not uncommon, and some countries lack these facilities or treat wastewater only partially. Rosa and Clayson, 2010, and in many occasions, treatment is not enough to remove all the contaminants. Paul and Meyer, 2001.